Hey everybody, today we're going to be covering section 8.12 and 8.13 in the same set of notes. We're going to be covering LD50 and dose response curves. So learning objective for today is that you can define the lethal dose 50%, which is known as LD50, and the learning objective is also that you can evaluate dose response curves. So the essential knowledge that you're going to gain is what LD50 is and what a dose response curve is as well. So we're first going to look at dose response studies and LD50. So these studies are going to be studies that expose an organism to different doses, so different concentrations of a chemical in order to measure the response, the effect that it has on that organism. So when they're setting up this experiment, the independent variable is going to be the concentration of the chemical, the dose, which is going to be added to water, to food, or air, something like that. And then the dependent variable is going to be the response that's measured in the organism, usually death or some sort of impairment. So that's why it's called dose response. We give them a dose and we see the response. Now, LD50 is a specific term that this is basically a measurement of the dose or concentration of the chemical that will kill 50% of the population being studied. So for example, arsenic's LD50 in mice is going to be 13 milligrams per kilogram. So a mouse isn't going to be a kilogram. That's a lot. But basically, if you were to take the smaller amounts, like maybe convert it to grams and all of that. But either way, essentially, if we were to take the mass of our mouse and within its body, it has 13 milligrams per kilogram, that is going to be the amount that half of the mice in our study is going to die. So that's why we call it arsenic's LD50 in mice, this is the amount that we would have to have within that organism in order to kill 50% of the organisms in our study. Now, LD50 data can be expressed kind of in a few different ways. Sometimes it's going to be mass, so like grams or milligrams per body unit mass, kilogram, like the mouse example I just gave you. Or sometimes it's going to be parts per million, which often is going to be in like air or water. Or we can also have mass per volume, which is typically how we're looking at it when it's in water or in blood. You can see it in different ways, but again, essentially, LD50 is where we have 50% of the organisms dying when we're at that concentration. So what is a dose response curve? Now, this is going to be a curve where we are graphing the data, where we are going to have the percent mortality on the y-axis and the chemical concentration on the x-axis. And it's going to look like this. So on our x-axis here, we have the dosage, and then on the y-axis, we have the percent mortality. Now, a lot of times we're going to be getting this data in a dose response curve, so that's why I want you to know how to look at it. The lowest dose where we have any sort of effect is going to be called the threshold or the toxicity threshold. So we're going to start having some effects like down here. And then we are also, so if you look at here, like on our graph, it's going to be 40 milligrams per kilogram. That right here is going to be your threshold where we start to get some data. And then we have the dose response curve. The whole curve itself is usually going to be S-shaped. So we are going to have lower mortality at these low doses. We're going to have higher mortality at the high doses, and it's going to level off at about like 100% mortality. Because if you get so high, eventually they are all going to die off. Now, LD50 is going to be this 50 dose where we get 50% mortality. So for example, on this graph, it's going to be at 100 milligrams per kilogram because we've got the 50 here, we've got the 100 there. Where it meets is going to be at this dosage. So that's where our LD50 is. And usually with this S shape, your LD50 is going to be kind of right about in the middle of it. Well, it will be in the middle of it because that's where the 50% is on the y-axis. So some other terminology that we have is ED50 and then other dose responses. So ED50 refers to the dose concentration of a toxin or chemical that causes a non-lethal effect. So it's like our effect 50. So things like infertility, paralysis, cancer, etc. They aren't dying but they're not doing so great, and we are having an effect on it. And again, the 50 refers to 50% of the population. So if like 50% of the population gets cancer from something, that's going to be the ED50. They aren't dying, but they're still having an effect. So for example, like the concentration of atrazine in water that causes 50% of frogs to become infertile, that would be like our ED50 for atrazine. And we're going to see the same general S shape that we have in LD50 dose response curves. It's just going to be at lower concentrations because it's going to not have so much toxicity. You're just going to have like lower amounts where you start to get the bad effects of it. So that's going to be ED50. But again, the graph looks about the same. It's just going to be like lower amounts. So how do we get this dose response data with human health? Now, obviously, it's very problematic to be giving a lot of toxic things to humans and then seeing how many of them die. We can't do that. Science is against that. So instead, we have to do this on animals. So this data is going to be collected usually from mammals like mice or rats because we are mammals, and they're used to simulate human toxicity. So we are getting this data from non-humans, and then we're extrapolating it to humans, which sometimes you wonder about how much it does, but scientists do a lot of work to make sure that it's as valid as it can be. Basically, the way that we do this is to determine the maximum allowable level for humans. So when we say like, hey, this pesticide is safe, or this amount of toxin in water is going to be safe, generally we're going to take the LD50 or the ED50 from animals, and then we are going to divide that by 1,000. Just to have extreme caution. So theoretically, some of our human LD50 amounts could be way higher. We just like take it down to like the smallest amount just to be careful in case it's something that we are more susceptible to, or just because you don't want any risk of 50% of people getting sick from it or dying from it. So the way that we do these studies, they're broken down into two types of studies. It's called an acute study versus a chronic study. Most dose response studies are considered acute, which means they only measure effects over a short period of time. They're also going to be isolated in a lab, so they don't measure like ecological effects of organisms dying, the trophic cascades, which is why we said DDT was safe and then come to find out it was not safe at higher trophic levels, because a lot of these studies are done in like small, short periods of time, isolated labs. They are not chronic studies usually. Chronic studies are going to be longer term, and then they follow developmental impacts. So for example, we study all the way from hatchlings to sexual maturity. And so although we take like huge precautions by dividing these concentrations by 1,000, trying to do like the smallest allowable amounts for humans, it's important to recognize that like the dosages that we have, and when we're testing like pesticides and other pollutants and saying it's safe, it is based on our knowledge. And usually we eventually have chronic studies that are going to go and look at things, and a lot of pesticides, they then approve it and then they study it for decades and find out, yep, we were right, there aren't any problems. But you just have to kind of keep in mind for some of these safe amounts, like a lot of our studies are happening on rats in labs over a short period of time. And so it's important to kind of be aware of that when we're looking especially at like newer technologies coming out. And this is part of why some people have concerns and they want to be like totally organic and they don't want pesticide use and all of that, because sometimes they worry, all right, what are we going to get the data from chronic studies? Now, keep in mind, if a chronic study comes out that starts telling us it's going to cause cancer in lots of people, EPA will pull it. It's just important, especially for emerging technologies and things, to be aware acute versus chronic studies and how these dose responses are usually done. All right, here's going to be a practice set for you, and you're going to practice a couple skills of data analysis and using some math. So we are going to look at this data table right here. And this data table is showing us the results of a dose response study for polonium toxicity in rats. So over here, we've got like the RTI number, and then this is going to be like our, it's like the type of rats or the group of rats. We've got our ED50 here, or LD50 here, TI, which is going to be LD50 divided by LD50, and then the minimum lethal dosage of polonium in milligrams per kilogram. So anyways, I want you to look at this data, and I want you to explain how this data could be used to determine a maximum allowable level for humans. And then I want you to identify the lowest dose that triggered a response in the RTI number 336 rats, and the dose that resulted in 50% of the RTI 336 rats dying. So you're going to have to do a little bit of extrapolating from this data to answer these. And that's your practice FRQ for 8.12 and 8.13. And those are also your notes on 8.12 and 8.13.